Welcome to another episode from the Veterinary Medicine Digest. Today, we are taking a deep dive into one of the most enigmatic and traditionally misunderstood structures in the entire vertebrate brain, the cerebellum. For a long time, the name cerebellum, which literally translates to little brain, reflected a certain scientific dismissal of its complexity. We were taught in school that the cerebellum was the seat of coordination and balance, the silent partner that simply made sure your gait was smooth and your reach was accurate. However, recent breakthroughs in both human and animal research are rewriting this narrative completely. We now know that the cerebellum is a powerhouse of information processing that reaches into the realms of emotion, cognition, behavioral timing, and even personality. To understand where we are today, we have to look at how our understanding of this little brain has evolved. In the early 19th century, pioneering researchers like Jean-Marie Pierre Flourens used experiments with pigeons to show that damaging the cerebellum resulted in a loss of flight and wing coordination. This led the scientific community to solidify the view that the cerebellum was purely a motor control center, a view that held strong for over 150 years. But there were always whispers that something more was going on. Even before those pigeon experiments, some researchers were curious if cerebellar size correlated with intellectual capacity, and by the mid-20th century, electrophysiological studies began to show that the cerebellum had complex connections to the limbic system, which is the emotional heart of the brain. The real shift in our understanding, the one that paved the way for the clinical canine studies we are discussing today, began with the work of Jeremy Schmahmann in the late 1990s. He proposed a concept that initially met with significant resistance, the cerebellar cognitive affective syndrome. Through careful clinical observation and functional neuroimaging, it became clear that damage to the posterior parts of the cerebellum in humans did not just cause a drunken gait or tremors, it caused significant impairments in executive function, language processing, and emotional regulation. Patients might become irritable, disoriented, or show a distinct flattening of their emotional responses. This discovery led to the idea of functional topography, which is the notion that different regions of the cerebellum are specialized for very different tasks based on their anatomical connections to the rest of the brain. In humans, functional magnetic resonance imaging has revealed that the anterior lobe, the part toward the front, along with parts of the sixth and eighth lobules, is primarily concerned with sensoromotor functions. This is where the classic motor maps of the body are located. If you move your right hand, you see activation in the right side of the cerebellum in these specific lobules. However, when you move to the seventh lobule, including regions known as crus 1 and crus 2, you enter the supramodal or cognitive zones. These regions are functionally connected to the prefrontal and parietal cortices, areas of the brain that handle the heavy lifting of thinking, planning, and memory. When humans perform complex linguistic tasks like generating verbs or reading, these posterior cerebellar regions light up. Even emotional processing, such as viewing images that evoke sadness or grief, activates the posterior vermis, which is the midline structure of the cerebellum. This functional topography is not just a human phenomenon, we see it mirrored in fascinating ways across the animal kingdom. Take, for instance, the humble goldfish. Researchers have used the optokinetic response in goldfish, which is the involuntary eye movement used to stabilize vision during motion, to study how the cerebellum predicts the future. By using periodic visual stimulation, they discovered that goldfish can actually learn to predict when a movement should start and when it should stop. If the fish is trained with a visual stimulus that moves back and forth at a regular interval, it begins to slow its eye movement before the stimulus even changes direction. This predictive timing is entirely dependent on the cerebellum. If you remove the cerebellum from these fish, they lose the ability to acquire or maintain these predictive eye movements, even though their basic ability to follow motion remains somewhat intact. This suggests that one of the fundamental jobs of the cerebellum is to act as a time machine, using past experiences to predict the timing of future sensory events. Moving from fish to mice, we find even more detailed evidence of how the cerebellum initiates movement. In one remarkable study, mice were trained to push a lever in response to an auditory go cue to receive a water reward. The researchers used advanced imaging and electrophysiology to look at the activity of neurons in the motor thalamus that receive direct input from the cerebellum. They found a very specific pattern right before the mouse initiated the push. There was a massive time-locked increase in activity in these thalamic neurons. This signal was temporally uncoupled from the auditory cue itself. It was a pure motor timing signal that dictated exactly when the movement would start. The study went a step further by using light-sensitive proteins to either block or trigger this pathway. When they silenced the cerebellar output to the thalamus, the mice were physically unable to initiate the learned push movement, even though they were clearly alert and interested in the reward. Conversely, when they used light to stimulate the pathway, they could actually trigger the lever push movement in the absence of any auditory cue. But here is the truly incredible part: this trigger was context dependent. If the mouse was in its usual training setup with the lever, the stimulation triggered the learned push. But if the mouse was moved to an altered environment with no lever, the same stimulation triggered a variable, unlearned limb movement. This shows that the cerebellum provides the when of the movement, the precise timing signal, which then converges with other parts of the brain that provide the what, based on the animal's environment and goals. Further research in mice using a licking task has shown that Purkinje cells, which are the main output cells of the cerebellar cortex, also ramp up their activity before both the initiation and the termination of a movement. Interestingly, this ramping happens much earlier when the movement is self-initiated rather than triggered by a cue. This supports the idea that the cerebellum is deeply involved in the internal preparation and timing of repeatable actions, effectively organizing the temporal structure of voluntary behavior. These fundamental mechanisms help explain the complex clinical presentations we see in our canine patients. One condition that has long been associated with the cerebellum is idiopathic generalized tremor syndrome, which used to be called white shaker syndrome. This name was actually a bit of a misnomer, as a large retrospective study of 75 dogs found that this syndrome affects dogs of all coat colors and many different breeds, though smaller, younger dogs like West Highland White Terriers and Cocker Spaniels are often overrepresented. These dogs present with an acute onset of rapid, low-amplitude tremors that get worse when the dog is excited or stressed. While we often describe this as a cerebellar disease because it often comes with other signs like ataxia or a head tilt, the tremors themselves are difficult to explain through a purely cerebellar lens. Histopathology in some cases has shown that the inflammation in these dogs is actually diffused throughout the central nervous system, not just limited to the cerebellum. This highlights how the cerebellum acts as a hub. When it is inflamed, it can disrupt signals throughout the brain, leading to that characteristic full-body tremor. Most of these dogs respond remarkably well to corticosteroids, which supports the idea that this is an immune-mediated inflammatory condition. Interestingly, some of these cases are preceded by gastrointestinal signs, leading some to wonder about the gut-brain axis and how systemic inflammation might target these specific cerebellar pathways. We also see unique cerebellar presentations in dogs with congenital malformations. There have been reported cases of a transient postural vestibulocerebellar syndrome in dogs with suspected cerebellar hypoplasia, which is where the cerebellum fails to develop fully. These dogs might appear almost normal most of the time, showing only very mild coordination issues. But then, something specific happens. If the dog extends its neck to look up or sniffs the ground in a certain way, it is suddenly hit with a massive wave of vestibular signs, including severe head tilts, vertical nystagmus, where the eyes flick up and down, and an inability to stand. This lasts for maybe 15 seconds, and then the dog goes back to its normal self. This happens because the part of the cerebellum that handles the vestibular system, the vestibulocerebellum, is the part that is underdeveloped. This region is supposed to constantly inhibit and control the vestibular nuclei in the brainstem. When the dog changes its head position, it puts a stress on the vestibular system that the malformed cerebellum simply cannot compensate for. For a few seconds, the vestibular nuclei are disinhibited and run wild, causing those dramatic symptoms until the system manages to settle back down. This is a perfect example of how structural changes in specific cerebellar regions lead to highly specific, often episodic, clinical signs. Now let us turn our attention to the main feature of today's episode, a groundbreaking multi-center study that finally brings the concept of functional topography directly into the veterinary clinic. This research analyzed 102 dogs with cerebellar disease to see if they could correlate the location of lesions on magnetic resonance imaging with the specific clinical signs the dogs were showing. This is the kind of lesion symptom mapping that has been so vital in human medicine, but was largely missing for our canine companions. The researchers used the vitamin mnemonic to categorize the underlying causes of disease in these dogs: vascular, inflammatory, traumatic, anomalous, metabolic, infectious, neoplastic, and degenerative. They found that vascular disease, specifically ischemic strokes, was actually the most common diagnosis in their group, followed by neoplasia or tumors, and inflammatory conditions. They also found that they could optimally differentiate between these causes based on age and the speed of onset. For example, if a dog was older than 75 months and the signs came on in less than 2 days, a vascular stroke was nearly 15 times more likely than other causes. Dogs with strokes were often non-ambulatory and were much more likely to be vomiting. On the other hand, inflammatory and infectious conditions like idiopathic generalized tremor syndrome were the only ones strongly associated with full-body tremors and signs of pain. But the real magic of this study was in the topography. By meticulously dividing the cerebellum into its various lobules and nuclei, they identified 3 distinct region-related cerebellar syndromes in dogs that closely parallel what we see in humans. The first is the cerebellar motor syndrome, which is linked to the rostral cerebellar lobe, the part of the cerebellum located toward the front and top. This area is considered a pure sensoromotor structure with direct connections to the motor cortex of the forebrain. The researchers found that lesions in the rostral culmen were significantly associated with cerebellar ataxia, that classic uncoordinated walk. Even more specific was the finding that lesions in the lobulus centralis, which is at the very front of the rostral lobe, were strongly linked to decerebellate rigidity. This is a dramatic posture where a dog's neck is arched back and its front legs are extended straight out and very stiff. This happens because the rostral lobe normally provides essential inhibition to the muscles that fight gravity. When that inhibition is lost, those muscles overcontract. The second syndrome they identified is the cerebellar behavioral syndrome, and this one is a real game changer for how we think about dog behavior. This syndrome is linked to the caudal lobe, the part toward the back, specifically the regions known as the folium and tuber vermis. Dogs with lesions here were much less likely to have severe motor issues like the inability to walk. Instead, they showed abnormal behavior. They appeared disoriented, subdued, lethargic, or sometimes hyperreactive and anxious. Owners would often report that their dog was simply not themselves or seemed reluctant to play and exercise. This is remarkably similar to the cognitive affective syndrome in humans, suggesting that the back of the canine cerebellum is responsible for regulating emotion and interaction with the surroundings. If a dog comes in acting weird but walking okay, we can no longer just assume it is a primary behavioral issue. We have to look at the caudal cerebellum. The third and final syndrome is the cerebellar vestibular syndrome, which the study localized to the deep white matter, specifically the area of the interpositus nucleus. This region was significantly associated with the classic vestibular triad head tilt, pathological nystagmus where the eyes flick involuntarily, and vestibular ataxia where the dog leans or drifts to one side. Interestingly, they found a strong link between lesions in the interpositus nucleus and paradoxical vestibular syndrome. This is a confusing presentation where the dog's head tilts toward the side opposite of the lesion. In a normal vestibular case, the tilt is usually toward the lesion, but because parts of the cerebellum normally inhibit the vestibular system on their own side, a lesion there causes overactivity on the side of the lesion. This makes the brain think the head is turning toward the lesion side, so it compensates by tilting the head away to the good side. The researchers also noted some interesting findings regarding postural reactions, which are the tests we do to see if a dog knows where its feet are. We used to think that cerebellar disease should not cause a delay in these reactions, that it should only make the movements exaggerated or clumsy. But this study found that delayed or even absent postural reactions were actually the second most common finding, and they were strongly associated with lesions in that same deep white matter near the interpositus nucleus. This suggests that the deep parts of the cerebellum play a much more direct role in proprioception, the sense of body position, than we previously gave them credit for. One limitation the researchers faced was that many of their cases involved vascular strokes that affected multiple regions at once, which can make it hard to pin a single symptom to a single spot. However, their large sample size and sophisticated statistical analysis allowed them to pull these specific patterns out of the noise. They also noted that because there is not a standardized way for veterinarians to describe eye movements or personality changes in dogs, some findings might have been underreported or misclassified. They suggested that using things like the Canine Dementia Scale or more standardized cognitive tests could help us understand the behavioral syndrome even better in the future. So, what does all of this mean for the everyday practice of veterinary medicine? It means that our neurological examination is more powerful than we thought. We can no longer just say this dog has cerebellar disease and leave it at that. By looking closely at the type of signs, we can begin to predict exactly where the lesion is and what the most likely cause might be. A stiff, rigid dog with a drunken gait but a normal personality likely has a rostral lesion, perhaps a stroke in the rostral cerebellar artery. A dog that is acting anxious and disoriented but can walk fairly well probably has a caudal lesion, which might be more indicative of a tumor or an inflammatory process, and a dog with a dramatic head tilt and eye flicking likely has deep white matter involvement. This research also gives us a new way to talk to owners. When a dog has a caudal cerebellar stroke and starts acting depressed or angry, we can explain that this is not just a reaction to being sick, it is a direct consequence of where the brain was damaged. We can tell them that their dog has a behavioral syndrome that we expect to improve as the brain heals, just as we expect the gait to improve. It also highlights the importance of not giving up on these dogs, even when they are non-ambulatory and vomiting from a massive stroke. The overall prognosis for cerebellar vascular disease is often quite good with supportive care. As we move forward, the little brain continues to reveal its secrets. We are learning that the uniform structure of the cerebellar cortex actually hides a profound functional diversity. This is known as the universal cerebellar transform. The idea is that the cerebellum performs the same basic computation on all the information it receives, whether that information is a motor command for a limb, a linguistic sequence for a word, or an emotional response to a friend. It takes that information, predicts the timing and the outcome, and provides a correction signal to the rest of the brain to make the output more precise, more appropriate, and more efficient. We are even starting to look at how the cerebellum develops and how sensitive periods in early life can lead to long-term issues. Research into perineuronal nets, which are like tiny physical scaffolds that surround neurons and lock their connections in place, is showing that the cerebellum's ability to learn and adapt changes throughout a dog's life. Issues with these nets or with the development of specific cerebellar cells are now being linked to autism spectrum disorders in humans and social deficits in animal models. This could one day lead to new therapies that target the cerebellum to help animals with profound behavioral or social impairments. In conclusion, the cerebellum is far from being just a little part of the brain. It is a sophisticated, topographically organized hub that is essential for every aspect of what makes a dog a dog. It allows them to time their movements, to regulate their emotions when a stranger walks by, and to maintain their sense of balance in a complex world. The latest research is a major milestone in veterinary neurology, providing us with a roadmap to navigate this complex structure in our clinical patients. It reminds us that every sign we see on a neurological exam, every flick of an eye, every stiffened limb, every change in a dog's spirit, is a clue to the incredible processing happening inside that little brain at the back of the skull. As we continue to integrate these findings into our practice, we become better clinicians, better advocates for our patients, and more appreciative of the exquisite complexity of the animals we treat. The cerebellum may be small, but its impact is massive, and we are only just beginning to truly see it for the masterpiece that it is. Thank you for listening. The titles for the reference papers are in the video description. From VetMed Digest, where we keep minds inspired, hearts light, and tails wagging.